Now we'll switch over to here. That black screen coming. There it is. So again, this is the Hubbard model, like the purple plaque for the eye. This is the ear, and remember that with the ear, we've got three different sections to it, right? We have the external ear, we've got the middle ear, and then we have the internal ear, okay? So the external ear, you're going to have the auricle or the pinna, all of this that's out here. This is going to be the external acoustic meatus, or the external acoustic canal, or the external auditory. You can switch auditory for acoustic. You can switch meatus for canal, okay? But external is going to be pretty important, okay? So this is the external acoustic meatus or canal that we see going through there. And then that is the tympanic membrane, right? You used to be able to call it the eardrum. Now it's the tympanic membrane. That's our border between external and now middle ear. So after the tympanic membrane, in the middle ear, we have our three ear bones, the malleus, the incus, and the stapes that we see right there. And we also see the eustachian tube coming in through here. You can also call this the auditory tube if you want. This connects to the pharynx. So we see that there. Now we get to the inner ear. And remember, we have structures that are associated with equilibrium over here, structures associated with hearing over here. So this is the vestibule. Inside of the vestibule, we have what are called the utricle and the saccule that give us what's called static equilibrium. We'll talk about that in lecture coming up. And then over here, we have the semicircular canals oriented in the X, Y, and Z direction. And these give us what's called dynamic equilibrium, or also known as rotational, because there's a fluid in here that rotates around, that sloshes around when we kind of spin on teacups in Disney World or whatever we're doing. Right? So these are the semicircular canals. This is the cochlea. The cochlea is where the hair cells exist that allow us to sense sound. And then this is the vestibulocochlear nerve. And now we know that cranial nerve number 8, that name breaks down to the fact that we have some impulses coming from the vestibule and those associated with equilibrium, and we have impulses coming from the cochlea that give us the information about sound and hearing. Okay? So this is purple plaque. Again, we have a 3D version of the ear on the 3B model, if you will, which is the one that we have labeled online through the Southern Dobson website. It shows the same exact structures. Okay? The other thing that we want to talk about is the cochlear section model. So as you can see here, we've cut out a section of this snail shape or cinnamon roll shaped cochlea. And as you would think in a snail, there's going to be a coiling tube that goes around and around. But what we'll find is that in here, there are actually 3 tubes that are going around and around and around. So we're going to look at just one section of one of these coils going around. So we're going to zoom up on something like this in our cochlear model. I'm going to move this out of the way. Try not to trip on it or something like that. And now we move on to this. So this is one of those coils that's going around that we're zoomed up on. And now we can see there are 1, 2, 3 different sections of it, right? So those 3 sections are divided by 2 different membranes. You see one there, and we see one over here. So this is called the vestibular membrane. And so this tube right here is called the scala vestibuli. Okay, so the names go together. Vestibuli and vestibular membrane that we see there. This one's called the scala tympani, like tympanic membrane. And then the other dividing line that we have right here is called the basilar membrane. And the basilar membrane, to a certain extent, serves as a base for this all-important organ that we have right here. It's called the spiral organ, or it's called the organ of Corti. And this is where our hair cells are embedded that we see right here. So we see the hairs sticking up off of the hair cells that are right here. So the organ of Corti, of the spiral organ, sits on the basilar membrane that we see right there. This middle section right here is called the scala media, like middle to a certain extent. You can also call this the cochlear duct. But the last thing that we see is another membrane that's right here. It's called the tectorial membrane. And the tectorial membrane is almost like a hand that we see right there, where those hair cells are, the tips of them are embedded within. And so when the sound waves come in, what happens is it causes this basilar membrane to bounce. As the basilar membrane bounces, this whole spiral organ bounces, and those hair cells are going to be jammed up into that tectorial membrane. And when they bend, they will then send an impulse into the brain via axons that we see here. Okay? So these hair cells that we see here that allow us to detect sound waves are actually mechanoreceptors. Right? So we have chemoreceptors, photoreceptors, thermoreceptors, nociceptors. Mechanoreceptors take a physical deformation, a bending of them, to open up their channels. And so this, with the tectorial membrane over it, is how these different hair cells of the spiral organ or organ of Corti are going to be able to send a signal into the brain. And this that we see here is our vestibulocochlear nerve. So we can see some of our bipolar neurons there. There's our different nuclei of these neurons headed in towards the brain. Okay, so three different spaces: scala vestibuli, scala tympani, scala media or cochlear duct, divided by two different membranes: vestibular membrane, basilar membrane. On the basilar membrane, serving as a base, we have the organ of Corti, or the spiral organ, with its hair cells embedded within the tectorial membrane that we see right there. And this yellow that we see here represents that vestibulocochlear nerve number 8. Okay? So there is an image of this online on the Red Mountain website as well. Since it is exclusive to our campus, we have the picture of it there. Okay? So make sure that you can identify those structures of the ear on both models, a 3B model as well as a purple plaque like I showed you. And then this is our cochlear section model. And those things that I pointed out are on that list that we have of extra things to know for Lab Practical 4. Okay? All right. That's what we needed to know.